Today, it is my honor to present to you Debbie Pittman and David Beidler from Pfizer, who will be presenting Forging a Path for Early Clinical Development in Sickle Cell Disease, from biomarkers to clinical endpoints.
Okay, thank you, and thank you for the opportunity today to speak about the work that we've been doing on early clinical development in sickle cell disease. David and I are going to be doing a tag team presentation covering a couple of topics. First, the part of the talk will give a very brief overview of sickle cell disease, so we're all on the same page as we move forward. And then we'll go on to talk about where early clinical development is in sickle cell disease, and then proceed to talk about some specific examples where we bridge non-clinical data to clinical data using an example of phosphodiesterase inhibitor PD9, and then the majority of the talk at the end will be focused on a phase 0 longitudinal study in sickle cell disease. Please feel free to ask questions as we're going through the talk. So briefly, sickle cell disease is a rare disorder. It affects approximately 100,000 patients in the US, but it's really a larger number of patients outside the US, greater than 2 million worldwide. It's a chronic condition with substantial morbidity and mortality and a clear unmet medical need. The life expectancy in the developing world is 40 to 50 years. However, in the US, it's between 40, outside the US, it's between 20 and 25 years. It's a very complex pathophysiology. There are currently 2 approved therapies for sickle cell disease, hydroxyurea, which was actually approved early in the 1990s, and it reduces these painful crises about 45%, but it does require extensive optimization of the therapy. There's poor compliance in terms of patients taking the hydroxyurea, and in addition, there's a black box warning. L-glutamine was recently approved. It's well tolerated. I think they're still out on what the clinical benefit is, but there is some uptake with the patients. The patients have these recurring pain managements, and the current therapy for that is primarily hydration and pain medication. So sickle cell disease is caused by a single point amino acid change at glutamic acid position 6 in the hemoglobin molecule. And what this does under conditions of hypoxia, it causes the globin chains to polymerize. And this forms the long fibrils, and what this results in is changes in the cell membrane. And that's what gets the typical sickle cell that you see and how it describes the disease. But it's not just the sickling of the cell that causes problems. The membranes can flip, exposing phosphatidylserine, which can impact the coagulation system. There's hemolysis, which actually contributes or releases heme, and that contributes to the inflammation. The cells also become sticky. They stick to the endothelium. They form heterotypic aggregates, and what you get is a vascular blockage. And it's this vascular blockage that causes a vaso-occlusion, and patients undergo these painful crises that sometimes requires hospitalization, and you'll see from some of our later data how patients choose to treat these painful vaso-occlusive crises. But the vaso-occlusion also causes other aspects of the disease. There's downstream complications. There's end organ failure. There can be an acute chest syndrome. There can be stroke. There can be skin ulcers, as well as osteonecrosis. So there's clear unmet medical need, a very complex pathophysiology when you're trying to treat, and you don't want to just treat the VOC, but you want to get to the root cause of the disease and understand what the patient's need is in terms of effective therapy.
So now we're kind of setting the stage, and we've been using, and actually those Pfizer people, and this is Nick's Clark idea. So it's a nice visual for what we feel. The current state of sickle cell clinical trials and recognition by the medical community and treatment really is the tip of the iceberg. There's a lot more that we're going to talk about. The clinical endpoints are about medical utilization. A crisis is only a crisis when you go to the ER or hospital and get admitted. We feel that's obviously just a small fraction of what's going on. Length of hospitalization stay or IV opioid use have really been used in interventional studies and are really regarded within the established community as meaningful issues with the sickle cell disease, as well as some of the end organ damages. Whereas what we do know from interviewing patients and from their perspectives, there's a lot of things going on underneath the water. And we have things like at home, their fatigue, their quality of life, They can't get up, they can't go to school, they can't do their work, they can't have their social life be the way they want it. They're medicating at home. They have anywhere from massages to relaxation to yoga, anything to try to relieve the pain and reduce the onset of these crises. So again, their daily life is severely impacted. And underneath all this, of course, there really are no validated or even really qualified biomarkers for sickle cell disease. There are a lot of studies that have looked at before and after and try to understand what is elevated in sickle cell relative to healthies, but nothing really that the regulatory agencies recognize at this time. So again, a lot of issues here around building in things at home and in the outpatient that just aren't being recognized in the clinical studies or validated in the medical community. So in addition to knowing that, my role a lot in the precision medicine and the translational part is we try to learn early in our early clinical development as early as possible. We want to build this value of confidence index that we call it. Just a nice way of saying that we think we're testing something that is meaningful. And the earlier we learn, the better. In the traditional system, you would put a drug into the clinic. You would do PK and tolerability. Is it safe? Is it kind of acting the way you want? And you really only learned maybe when you hit your clinical endpoints, your proof of concept. But between there's a very low confidence, and if you're relying on hope, and you're kind of putting it off, saying we hope to hit those POC endpoints, because it's safe and tolerable, but you may get it in several years in and be disappointed. And Pfizer and other companies have come up with a lot of strategies to build in early confidence around understanding natural history, the patient perspective, and a lot about biomarker criteria. And anywhere from that you're engaging the target to modulating a pathway or a mechanism, all the way to is it really relevant to a disease. And that's why we have some milestones around either proof of mechanism or something analogous to a surrogate signal of efficacy. The earlier in the clinic that you can learn, even in healthy volunteers, that your pathway is engaged, the more confidence and more value you're providing to your asset. And that can be very important in the competitive environment, both within your company and outside of your company. So the patient perspective, the need for this type of early value really kind of drove some early questions. And again, the schematic here that we're looking at is, in addition to the PK, by using in vitro assays and then get translated into the early preclinical models, are we engaging our target? Are we hitting a pathway? Does that pathway then feed into disease? I hope it does. That's your confidence and rationale is your pathway should have some relevance to the disease. Hopefully those overlap quite a bit. And of course then, how does that relate to the clinical endpoints? And especially in the rare disease area, there's usually very little data available linking all this together. So we try to build as much of this in place prospectively so that we actually have not just let's do some biomarkers and let's see what happens and hope for the best. We try to actually create some criteria and make it a little bit of a push so that our management or competition sees we're setting to do this. This is important to us. So very early on in the phosphodiesterase inhibitor, I'm going to have Debbie introduce this asset.
Okay, so in the next few slides, I'm going to focus on the non-clinical data around the PDE9 inhibitor. As I mentioned earlier, hydroxyurea is used in the clinic, and its primary mechanism of action is to increase fetal hemoglobin to shift the polymerization during that hypoxic state and therefore reduce the sickling. But hydroxyurea probably has other activities. It's anti-inflammatory. It can also act as an NO donor, and it can impact the soluble guanylyl cyclic pathway. Within this pathway, there's also an enzyme called phosphodiesterase 9, or PD9, which is responsible for the degradation of cyclic GMP to GMP. It's normally expressed in the brain, but also in sickle cell patients, it's elevated both in neutrophils as well as erythrocytes. And it's thought that this elevation leads to a degradation of the cyclic GMP and contributes to downstream activation, such as an increase in the selectins through the inflammatory process on the endothelial cell, but also an increase in the cellular aggregates through the white blood cells, and this would lead to an inflamed situation within sickle cell patients. So our working hypothesis was that inhibiting phosphodiesterase 9 could mediate some of these downstream events in terms of cellular aggregates as well as inflammatory markers. We routinely use two non-clinical mouse models in our studies. One is a TNF-induced inflammatory model in wild-type mice, and that's really just induced a general inflammatory response. but we also use a model that's a sickle cell mouse model, the Towns model of sickle cell disease. And this is a transgenic mouse model in which the globin genes of mice have been removed and the human mutated globin genes have been introduced into the mouse. And this mouse model actually exhibits much of the pathophysiology that's observed in human sickle cell patients. So in part of our studies with the PD9, we wanted to look at the effect of both short-term administration and chronic administration in both of these mouse models and look for effects on pharmacodynamic markers that might actually be translated to be used in early clinical trials. If you look on the left-hand side, following a chronic dosing scheme, here we can see compared to the vehicle or the dosing of the PD9 alone, we do see a decrease in plasma-soluble E-selectin, and in combination with hydroxyurea, we actually also see an additive effect. We've also looked at this in terms of looking at neutrophil platelet aggregates as well as cellular aggregates, and that's shown on the right, and I'll go into a little bit more detail in the next two slides on how we assess aggregates in the mouse model. We use intravital microscopy to look at cellular aggregates. In addition, after we've taken blood, afterwards we take blood for analysis of markers, but in this, what we do is we expose the cremaster muscle and we have specific antibodies that recognize neutrophils as well as specific antibodies that recognize platelets. So we administer these antibodies to visualize cellular aggregates and adhesion, and then we can do quantitative analysis of this. You can see on the left hand, when you look at vehicles, you can see that in this sickle cell mouse model, there's poor flowing of the blood. You see large aggregates flowing, as well as adhesion to the endothelium. As you add, in this case, the PD9 inhibitor, you can see that the blood flow has increased, as well as the cellular aggregates have decreased. And we also see that in combination if we use the PD9 inhibitor with hydroxyurea. We can then go on to quantitate this, and we generally look at 25 to 35 venules within a mouse, and then we do a quantitative analysis where we can see that administration of a PD9 inhibitor, we see a decrease in neutrophils adhered to the endothelium, but we also see a decrease in neutrophil platelet aggregates with the PD9 inhibitor alone, as well as in combination with hydroxyurea. So based on these studies, we would look at neutrophil or monocyte platelet aggregates in an early clinical study, as well as monitor some of the soluble factors such as soluble E-selectin, P-selectin, based on the data that we've gotten from the non-clinical model to try to identify potential pharmacodynamic models in the clinic.
That's where I pick up. So as the PM lead, my job is to try to take a lot of the findings from Debbie's group and try to make them feasible for going into the clinic. So we had a little bit of data from a pan-selectin inhibitor, GMI 1070, rivapansel. They had published a pilot model, a study, where they actually dosed stable sickle cell subjects. They weren't undergoing a crisis, but their basal levels of activity were there. And when they dosed their inhibitors and then collected samples at the 4, 8, 24, and 48 hours, they did see a movement in a panel of biomarkers that were relevant to the pathophysiology that Debbie just described to you. And while they all aren't included here, what was notable for us was that with this addition of this inhibitor in the stable subjects, they did see a reduction in the soluble E-selectin and P-selectin, as well as the ICAM. So again, an more anti-adhesive property was chronically produced with administration. And while they didn't look at the neutrophil in their monocyte platelet aggregates, they in fact did see a reduction as well. So I think you're seeing where I'm heading with this. We're starting to see some nice translation from some of Debbie's group's work in the Towns model to some early pilot studies with a drug that is a similar mechanism of action. And so the big question that came for our phase 1b study, which I'm going to mention in a minute, is can we generate a similar biomarker in the stable state? And just, again, this is about getting that mechanism in place. Can we create a less adhesive, less inflammatory state that might then have an impact on these VOC crises? So this is where Sanguine comes in, of course, that's why we're here today. So we needed patient samples, and we just didn't need a bunch of samples one at a time. We needed longitudinal patient samples, because we don't do drug studies with just one dose and one day and then let them go. You need to understand the longitudinal nature of these changes. So we were able to have, Sanguine was able to recruit 43 subjects through their mobile phlebotomy groups, and we were able to bleed them about every 2 weeks or so apart, so that we had, as you can see below, we had a panel of of longitudinal data from, I think it was about 36 patients, at least 2 to 5 or so apart. And now you're giving your statisticians really good longitudinal data. They can do a lot of work with that to understand things around, do you in fact have a longitudinal variability that's acceptable? And can you now predict and can actually estimate a cohort size moving forward into the phase 1B that will at least have statistical significance? And we will use some of the data from the pilot study and from Debbie's work to understand what is biologic significance. If it's going to take 80 or 90 subjects because of high variability, that may not be feasible. The good news was when our statisticians looked at some of our longitudinal data, they were able to in fact show us that we didn't need large cohorts, that we could get some answers about significant movement in some of these biomarkers with some fairly small, under 20 cohort sizes. So just very briefly with the setup of the study design, we did 5 mg BID and 25 mg BID, and again, that's twice daily dosing. We had 4 weeks of treatment, and then we had a 4-week follow-up. So I'm going to show you some biomarker collections kind of before, during, and after that active treatment period for these 2 doses. So what I wanted is, we did a very similar panel of biomarkers that have been done preclinically and in that pilot study. And what you see here is that, for example, with the monocyte platelet aggregates, at the end of treatment, at day 29, we did achieve some statistically significant reductions in the aggregates. Now, there is a lot of noise, but again, we were able to achieve the significance, and we saw roughly about a 40% reduction from where they were pre-treatment. Fewer aggregates, and think of those videos if they were a human video, that you have fewer of these aggregates floating around and flowing through the cells. Would that potentially be more conducive to avoiding these occlusions in the microvasculature causing the crisis? Same thing with the neutrophil platelet aggregates. We did see, again, roughly 30 to 40%, again, a little bit of a dose response here. Very small, so again, small study, quick answers. We're trying to get with a phase 1B study that we're getting our mechanism engaged. And even here with E-selectin, we're reducing soluble E-selectin levels. Again, endothelial activation is a little reduced. So reduced activation, reduced aggregation, is that now a more conducive state to avoid these VOC crises? So that's really was kind of the goal of the study, was that chronic administration, at least even in the stable state, because we're going for prophylactic use, they would take these drugs continuously, and hopefully this would then result in fewer crises in a given time period. So this is just to kind of sum up this stage of it. So what we've done with the translational part is we've put together from the preclinical models, the selectin and the aggregate data. We had some guidance from a clinical pilot study, and we now were able to fill in that bridging gap to say, yeah, with some chronic administration, we do see that similar type of profile that was seen preclinically and suggested clinically. So this really gave us guidance that as we now take on a larger study where we are going to look at crisis endpoints, which is much larger numbers of patients and more involved, we at least have some confidence that our dosing will at least be testing that mechanism. And that's really what we're trying to achieve with some of the early translational strategies. So that kind of ends chapter 2. Hopefully if there are any questions, let us know. But again, this is now setting up for taking this into the clinic and about clinical registration endpoints. Getting back to that iceberg, remember, we still haven't solved the iceberg problem. We still only have hospitalization and opioid use as a criteria. And so again, this was the definition. So the sickle cell pain crisis is no medically determined cause other than a vaso-occlusive event. Fortunately, we learned from interviews with our patients, they know when they're having a crisis and it's separate from their average daily pain. So they feel confident they can say that. But here, this is about actual medical utilization. And actually the duration here, depending on the clinical study that you looked at here, some required at least 4 hours, some were 2, and some were regardless. But again, this was all about medical facility visit that was required to kind of ring the bell that yes, you are having a crisis. You also then had some criteria around parenteral narcotic agents or NSAID agents while you're in the clinic. So again, this is all above the iceberg. Very much, again, it's about a care provider or caregiver-mediated, caregiver-centric environment, which it has some advantages. It's a controlled setting. You're in the hospital. You hope it's controlled, and then it was reported in other trials. It's familiar to the FDA. Again, it's well about precedent, and so this is what previous HU and other studies have used, and this is what and some of the cell G1 and other things they still were used some of the medical utilization. The disadvantages, on average, and even in the cell G1, they had a placebo had roughly 3 VOCs a year at most, generally 1 to 3 VOCs a year. So it's infrequent. A lot of it could be dependent on the patient's ability or willingness to go to the hospital setting. There is a stigma around sickle cell that I really want to touch on too much, but they feel hesitant to go to the hospital because they're treated as drug abusers, because they're coming in demanding IV narcotics. So there's that whole stigma. Are you really getting the true patient perspective? And it's really reflective of a meaningful clinical benefit. And again, that's where we're getting into. You really have to, if you're going to use medical utilization, how do you normalize access to medical facilities across all your investigator sites? That could be a challenge. Sure, you have a question?
Did you consider transfusion dependence?
So for this, for this type of control study, we would actually probably exclude the transfusion dependent phenotype because that changes the whole blood chemistry for us. But that's part of the equation. There's other standards of care that could confound your study here. So typically we would have that as an exclusion criteria. Great, okay. So we did have some background information. Wally Smith at Virginia had done a study in the early 2000s where again, the background was that it was, healthcare was a proxy for pain and the underlying crisis, and it really doesn't reflect the true number of VOCs that were being reported. So his group did a very nice study, again, following patients longitudinally and looked at the pain crisis. So it was basically 232 patients, and they did a paper diary, daily paper. This is early, this is before all this. I mean, smartphones were out, but they weren't where they are today. So we still had the old-fashioned stuff. So they had 31,000 days were analyzed by paper diary. They did see a lot of 56% of total patient days were filled with pain. When the patients reported a crisis, roughly 13% of the time that was managed at home, whereas only about 3.5% of the time it actually was being caused them to go to the hospital and seek medical utilization. So again, this bears out what we've been saying is that you're 3 times more likely to stay at home and manage your VOC. So you're now missing a large portion of an unmet need. These people are dealing with a crisis, but in an interventional study that would not be captured. So this was a problem to us, and we'll talk about that and more. So obviously more endpoints, quicker studies, smaller studies as well. So there's not just for the patient experience, but for the developer's experience. It helps us try to be more efficient and more effective in intervening and capturing that we're in fact creating clinical benefit. So we have a whole outcomes group, and we've been interviewing patients for a number of years now, and this really is about defining the patient experience. So they really want us to reduce the recurring VOCs. I think that's pretty obvious, but it's good to hear them talk about this. They'd love to be able to self-administer. They really have an issue with the fatigue components. They would really love to address the daily fatigue. Avoiding the stigma of medical seeking. They'd love to be able to treat at home. Again, all this preventing the long-term complications, improving their quality of life. They can be socially and active, go to work every day, go to school every day. And then really it's about the complication of the disease and actually being able to treat the root cause of the disease itself, not just the symptoms. So again, we're thinking of this disease now as a spectrum, and how do we cover activities at home, in the clinic, at the physician's office, at the ER, or actually in inpatient. So we're trying to capture the new endpoint that we're creating a new concept that's called the VOC day. And so we've kind of mapped out the conventional pain crisis on the left here and then our VOC day on the right. So both share the common thing. It's a sickle cell related pain crisis, and it's nothing medically determined cause other than a vaso-occlusive event. So that's common. But we now have, instead of relying on medical utilization or parenteral narcotics, we have daily reporting through e-diaries. So they self-report that they're having a vaso-occlusive pain episode. They also then have some secondary endpoints around daily pain, daily fatigue, their daily functionality, what medications are they taking, and do they in fact seek medical care. But this new VOC day really doesn't require that they go to the hospital to be registered as a VOC day or receive the IV narcotics, and duration really it's agnostic to duration at this point. This just kind of shows the idea of that the VOC day is still capturing the conventional day pain crisis that has been used in the previous studies, but in addition, we are trying to bring in these more patient-centric endpoints and create a tool that can capture these in a future interventional study. So as far as the overall patient experience, here's kind of the slide we use here's the concept of the patient experience. The various domains that we have are the actual VOC painful crisis, pain, fatigue, and functional impairment. The endpoints that we're using mostly through e-diary We ask them, we'll see in a later screenshot that Debbie's going to show you, are you having a pain crisis? And that you fill that out on a screen on your e-diary. But then every day, regardless of whether they're having a crisis or not, we're asking them about their pain, how fatigued they are, and what is their overall functioning. Each of these components, these were previously validated patient-reported outcome endpoints. So this is kind of a Frankenstein model. It's called a brief pain inventory. There's a fatigue scale that's been validated, and this is from a group called the Return to Normal Activity Questionnaire that asks about, can you do your self-care? Are you socially active as normal? Can you do your normal physical activities? And can you go to work or school? So this is, again, trying to get multiple concepts of the patient experience, because they told us it's not just about the crisis. They'd like to reduce the fatigue. They'd like to be able to be normally functioning every day. So how do we do this? Good question.
Are you using any electronic activity monitoring, like to see
Yeah.
how much sleep
Well,
and activity?
Yes, so we're going to show that in the ELIPSIS, because in addition to the background, we've been doing some actigraphy. Yeah. How this works then is, so they say they're having a crisis, and we also ask them, well, what are you doing about it? Where are you, or how are you then managing this crisis? So one of the questions that Debbie's going to talk about in a few minutes is, did you go to the hospital? Did you go to an ER or a doctor's visit? Did you just email or call the doctor, or did you really strictly manage this at home? So each day, even when they're in a crisis, we ask them, are you still in the crisis, and how are you managing it? So that we can create, when you now string together days, you create a VOC event. And so that can last anywhere from 1 to several days. And so if it's kind of what we try to show is, this is a hypothetical schematic of, if it's, for example, this patient on the very top started going, direct healthcare utilization, but then went home. Maybe they go right to the hospital. So we're learning more about this. Some of the early studies that we did with ELIPSIS, we then said, okay, we'll classify an event by the highest degree of medical utilization they had. So even if they just spent a day or so in the hospital, we'd still call that a hospital VOC. But you can see now that by monitoring this daily, you get some of that information you were asking about, is what is the daily progression of it? It's not just binary yes or no, in the hospital, out of the hospital. So this hopefully, while this, like I said, we still haven't worked this into a primary endpoint. You've got to kind of walk before you run with regulatory work. But at least we can do sub-analysis like this and see, maybe potentially you don't reduce the number of days, but you reduce the number of hospitalizations, or do the pain scores come down? So there's a lot of permutations you could look at from this type of analysis. So what we have now is, we have some initial biomarker data with PD9. We showed potentially a less adhesive, less inflamed state with E-selectin in the aggregates. We now had some tools in place, at least by talking to patients and building some tools around the VOC day. So we really then designed a study called the ELIPSIS study that really tied all of that together, getting ready to take PD9 and other drugs in our portfolio into interventional studies to look at vaso-occlusive crisis impact. So let me go back to our original iceberg. So now what we have is, we have daily EPROs, so that we're monitoring pain, fatigue, functionality, drug use, the daily activity. We are now introducing an actigraphy monitor if we want to see how they're moving on good days versus bad days and the transition from those days. And we're also then doing blood collections, longitudinal blood collections, which I'm going to talk about in a minute, that span the VOCs. We're trying to get blood before, during, and after these crises to see what is really going on in the circulation that is either causing or as a result of the VOCs. So of course, we continue the EPRO monitoring. We have the actigraphy. We do the blood collections, regardless of where they are. We did this at one site in Detroit, so we were able to follow these patients through their various levels of medical use. So it was a 6-month study with patients plus or minus HU. So we gave them, and this is where Sanguine comes in again. So they were basically our operations group on the ground in Detroit. They helped. Michael Callahan would recruit them in. We had phlebotomists giving them the actigraphy devices as well as the EPRO devices, and they would monitor daily pain, fatigue, and function and medication use. And then what we had was, there was a special cassette, which Debbie's going to go over on, for a VOC crisis. It would alert Sanguine and the clinicians that they were starting a crisis. Within 24 hours, we had a phlebotomist out to their house. The next day, we had a phlebotomist out to the house. 3 days later, we had a phlebotomist. Whenever it then ended, we had a phlebotomist out to their house if they needed to be, if they were at home. If they were in the hospital, we got it in the hospital. So this is what we're getting at here, is that we were really trying to get those longitudinal collections that spanned that VOC event. And so that we had both baseline data from when people were not experiencing a VOC, and then we had these VOC collections so that we could compare VOC versus non. And so we also had the continuous monitoring then with the Philips ActiWatch. Oops, I'm sorry. And then of course we had, if they weren't experiencing any VOCs, we just had collections every 3 weeks so that we could get a good baseline from them. So this just kind of points out what it is. VOC daily, actigraphy is going continuously. The black bars are the stable blood collections. And then when this hypothetical one had 3 or 4 VOCs, we had it day 1, day 2, and then 2 days after the resolution of the VOC. This is just showing kind of what our first question. So every given biomarker or every given PRO is going to have this kind of background noise. And what we wanted to see by looking longitudinally is, was there really a threshold where when we reported a VOC, did you actually see something that was statistically significant and also then clinically meaningful because it was during a VOC? So this is where we were as far as, this is what we're hoping to see, is that was it just going to be noise? If you ask a sickle cell patient daily, how's your pain? Our concern was it just might be chaotic. So hopefully we can convince you that we got a little bit of information about this that will really help us move on. So I'm going to stop my section here with just saying this was quite an operation because we did this in, Michael Callahan's group in Detroit recruited in the 35 or 60 subjects, but Sanguine did a lot of the operations. We had various vendors. We used Boston Children's Group for some of our aggregate assays, and that's Alan Michelson and Larry Freilinger. Really, we've been working with them for a number of years. Data management, all this putting together was a lot of work, and it took probably about at least 2 years or so to complete everything. But if you're going to put one of these together, it's not an easy thing to do. But it was really enjoyable, and hopefully what David's going to show you now it's providing us some really valuable information.
So this is just an example of what the ePro looked like and what one of the modules looked like. As David mentioned, it had 2 modules. There was the daily reporting, daily diary, which they entered their day, if they had a VOC day. A severity scale was adapted from the BPI. We had functional activity, fatigue, as well as a medication diary that they entered every day. There was also a button on here that they could push that said if they had a pain crisis. And what this did, we really didn't want to interfere with what their choice of treatment was. It did alert the physician, but it also alerted the mobile phlebotomist that they were having a pain crisis so that they could get blood drawn within 24 hours as well as 48 hours after the onset of a VOC. So these are the 2 modules of the how it looked on the first page of the ePro. And then this is just an example of what the patient chose for their treatment. In this case, they were asked, did you experience a pain crisis in the last 24 hours? If so, how long did that last? Did it last 4 hours or more? And then they could select what they decided for their treatment. Did they decide to treat themselves at home? Did they call or contact a doctor or a nurse via text or email or phone call? As well as they went to the doctor, they went to the emergency room, or they stayed in the hospital overnight. So this was all captured over the 6-month study for each patient. This is the demographics of the study. It was roughly a 50-50 mix of patients on and off hydroxyurea. We started with 37 patient volunteers. 2 did not complete the studies early on for various reasons. We also included the opportunity to include some non-sickle cell patients. We included sickle cell traits from the study, and many of these were from the same patients' family members. So we were able to include that. And we also had a few normals where we could get baseline data and use it for our comparison. The trait patients and the normals did not have, they only had blood draws for 3 months and they did not do the ePro. This is an example of what the schematics look like in terms of the longitudinal pain over the time in 2 of the patients. And you can see that the pain scale goes from 0 to 10. And in some cases, the patients would rate their pain as 10. We actually had some patients that had very high pain, and then there were other patients who would consider their daily pain low. So there is a lot of variability. But the advantage of a longitudinal study is we can see what that baseline pain is and then normalize it through the study. And then if you can see that the VOC is 4 hours, how long did the VOC last? Was it less than 4 hours is in blue? And then you can also see the VOC event. And you can see from the red that the patients that are having a VOC event or VOC day are experiencing the worst pain by their own pain scores. question? Yes.
From those two illustrative examples, it seems the compliance is pretty good for them to use this data.
Yeah, I think we had,
This is some cherry picking here, of course.
but we also had, was it 63%
With
compliance?
60 to 70% compliance for daily use, yeah.
Yeah.
There's a whole talk we could have about imputing data and how to fill that in.
Yeah, there was, as the study went on, you could see that compliance got less. Though we did have some learnings from this study. I will note that the ePro device was a separate device versus not on their phone. So when the patient would go to the hospital, or they wouldn't always remember to take the ePro device because they'll grab their cell phone. So there are some learnings from that you could take forward from this. It's probably best if you can get it on their phone because they'd always have that. This is just an example of what the VOC events and durations were from this study. The duration was an average of 3.25 days across the patients, as well as the annualized VOC events was between 7 to 8 events per year. You can see that some patients, there's one patient that actually had a fair number of events on this study. What was interesting, and this is really consistent with the Pisces study, is what you ask what the patients chose to do treatment. So if you look at where they were for a VOC day location, almost 72% of the patients chose self-treatment. They did not seek any medical utilization. That means if you're using the standard VOC definition now that requires medical utilization and an input from a medical a medical supplier, you're actually missing the majority of the patients and they're not seeking medical care. If you look at the VOC event location, it's roughly 65%. So really, a lot of patients there's a clear unmet medical need and they're suffering from their disease, trying to manage it through their own home pain management, hydration, massage, or whatever they choose.
So did all the patients have health care or was that captured?
These were actually, the patients did have health care these were all patients that were under care. If they chose, we didn't want to interfere with how they chose to treat their VOC.
I know it's a small sample, but did you see any differences in gender between self-treatment or also self-treatment versus going to the hospital location of their home relative to?
I don't think we saw any difference between gender. These patients were actually all fairly close to the Detroit Medical Center, so I don't think that actually played a factor in whether they chose treatment. Was there another question?
Yeah, I was wondering if you could comment on whether, if you have any hypotheses on why people chose to self-treat instead of going to a care center.
I think part of it is because they require, if you go to the emergency room, you often sit there for a long time, and then for some of the sickle cell patients, they're actually often viewed as drug users because they are seeking opioids. And some of it may be that all the medical centers really don't understand what the needs are for patients with sickle cell disease, that they really do need to have that pain treatment to reduce their crisis. And I think it's over time that it's probably easier for them until it reaches a certain point to treat at home. That's sort of our hypothesis.
But there were some that went right to the hospital.
Yeah.
So there is no one trend. And again, this group, Michael Callahan, somewhat, he recruited them himself. So these were actively seeking medical regular checkups. So these weren't even on the kind of the edges of treatment. It's hard to say. For whatever reason, this group, maybe if it's cultural, against anxiety, whatever, but maybe the self-treatment just is more effective too.
But as you're thinking about clinical trials and bringing new agents forward, it's really important to think about where the patient is seeking treatment and can you really start to help these patients that really choose not to go to the hospital or the emergency room to treat their VOC. This is looking at some of the secondary points where we looked at increased pain, decreased functioning, and fatigue. And here we're looking at normalized data from each patient to their baseline. And you can see, and we're looking at non-VOC days, which is in the blue, or the solid, And then the striped bars are all VOC days, and we tried to break it down to those patients that were at home or chose to go to the hospital. And I think in all cases you can see that in terms of increased pain, decreased functioning, and fatigue, when a patient is reporting that they're experiencing a VOC day, they are seeing an increased pain, decreased functioning, and fatigue. And we really didn't see any significant differences between whether they chose to treat at home or whether they chose to go to the hospital, depending on this analysis. We then also, as we mentioned, we had actigraphy. We used the Philips ActiWatch. And what this allowed us to do was actually the patient wore the watch all the time, and they could monitor their total daytime awake activity as well as their sleep activity. In the top panels, you can see what type of data that's collected from the individual points to kind of getting to more normalized data. So you can look at this data in a lot of different ways. I would say that it's a large amount of data to analyze, but you can look at activity counts per daytime activity, how sedentary they were there minutes of interrupted sleep, what a sleep fragment index was, as well as total sleep time. I would comment on the watch that the patients were also pretty compliant, but sometimes a lot of the patients were younger and sometimes on the weekends. You can see that the watch was somewhat, I would say, not attractive, and they would take it off on the weekends. So that's something else. As we move forward, we need to think about the type of actigraphy watch that we give to patients, that it's not something that they look at and say, I just can't wear this on the weekend or whatever. So this is where we look at changes in actigraphy, and we did see changes in actigraphy between patients that were reporting a self-treated VOC versus a non-VOC. You can see the black line versus the blue line. And not surprisingly, if you look at the healthcare utilization, you can also see that there's changes in terms of activity based on this analysis. And if you start to look at the percent change from baseline, you can see that there's a change in the total rest activity, as well as average activity daily, as well as you can see that there's differences in sleep when patients are reporting the self-reported VOC. We also had a large number of biomarker panels that we tried to incorporate into the study. So as David mentioned, we had regular blood draws during the whole time of the study to get baselines, but then we also had 24 hours, 48 hours after the onset of VOC, and 72 hours after resolution. We included a lot of samples that required either processing on site, such as the leukocyte platelet aggregates and microparticles, which required special processing and analysis within a certain time frame, as well as we had a microfluidics-based assay, which is a physiological flow-based assay, and that was done by functional fluidics to look immediately or within a certain period after the blood draw, like the same day, to see if there's any addition in adhesion to substrates such as VCAM1 or P-selectin. We had protein panels that included serum and plasma, cell adhesion markers, inflammatory markers, growth factors, as well as coagulation markers. And we've also banked samples. We've banked all the plasma serum samples, as well as we've banked samples for RNA analysis in the future, as well as DNA samples if we want to do profiling. We haven't touched these yet, but these are all samples that we'd hope to now analyze and that we would make available to others for analysis as well.
Can I ask quick? The visiting nurses, so we know that there's a lack of trust in the sickle cell community. How was that received by the patients?
That was actually received very well. The patients, like the mobile phlebotomist coming to their house and drawing blood, and I think they developed a level of comfort having the same person come to their home to draw the blood, as opposed to having to go into the clinic to have their blood drawn. Do you want to?
No, we heard back from our MDs. They felt they were really engaged and wanted to learn about the study, and were very at ease with those people coming to visit.
We had one patient that asked the phlebotomist to come in the hospital to draw their blood because they got. We couldn't do that, but it was. So this is looking at the cellular aggregates, and what we noticed, and this is consistent with what we've seen in the animal models as well as what we've seen in the PD9 study, that when a patient is reporting a VOC, we actually see an increase in cellular aggregates in terms of the monocyte platelet aggregates, as well as the neutrophil platelet aggregates when you look at baseline comparisons. In addition, we did see some changes in adhesion to P-selectin under flow conditions, shown in the bottom. So in both of these, we do see changes in aggregates when a patient is self-reporting a VOC. So in addition to the other biomarkers that we said, we also took clinical laboratories, and this is looking at the standard hemoglobin, hemoglobin F, nucleated red blood cells, eosinophils, as well as C-reactive protein. Interestingly, eosinophils actually decreased during the VOC, but one thing that we also consistently saw a difference in was the C-reactive protein. And this is actually consistent with a study that was done a number of years ago by Ballas, who actually did show some changes in C-reactive protein in sickle cell patients. And these would be very easily monitored in a clinical setting because these are part of the clinical lab monitoring.
Excuse me. Were those changes consistent if it was a hospital-based VOC versus a self-reported VOC?
We haven't seen differences between the hospital-associated and a self-reported VOC with the number of patients that we've looked at.
Again, very small numbers. Our statistician tried to parse them out. There's just not enough data. Yeah.
And then we looked at some cytokine and hemostatic markers, and it's not unexpected, but we did see changes in IL-6 from baseline, as well as interleukin-10, TNF-alpha, and as well as some coagulation markers such as TAT and a slight decrease in tissue factor. So based on this study, this electronic patient-reported outcomes, we believe, provided a more comprehensive accounting of the patient experience in sickle cell disease. The majority of the patients had at least 1 to 3 OC events during the 6-month period. Over 63% of these reported events, patients did not seek medical utilization. We did see significant differences in pain, functioning, and fatigue levels reported during the VOC compared to non-VOC, and the VOC reported days had higher pain scores, decreases in measures of functionality, as well as an increase in fatigue. We also saw differences in actigraphy, which reduction in daytime activity, as well as disturbance in sleep at night during a VOC. In addition, with the biomarker analysis, we did see changes during VOC days that were observed in a substantial number of clinical laboratory and biomarker measures. This included cytokines, hemostatic markers, C-reactive protein, as well as nucleated red blood cells, and we also saw changes in the monocyte and neutrophil platelet aggregates. This innovative at-home longitudinal study demonstrated the feasibility of employing and using mobile phlebotomy, electronic patient-reported outcomes, as well as actigraphy, and the feasibility of monitoring pain outside the hospital when a patient was experiencing a VOC. So the way that we're looking at sickle cell disease is that you have on the improved sickle cell disease where you have minimal endothelial activation, minimal numbers of cellular aggregates, and a decrease in that endothelial cell activation. As they go from sickle cell steady state to a VOC, you see the hemolysis of the red blood cell, you see inflammation triggers, as well as leukocyte adhesion. You also see an activation of the endothelial cell, increase in coagulation and thrombotic markers, and the cellular aggregates increase that vaso-occlusion. When we tried to do with our natural history study in sickle cell patients, as well as our preclinical models, is to take different markers of this, such as the clinical labs, plasma markers, physiological flow, coagulation markers, and then using EPRO and actigraphy to really try to get a better understanding of what might be going from the improved sickle cell state to when patients are experiencing a VOC. And David, turn it over to
For our
you.
final slide, so what does all this mean for us? And of course, we have to pay the bills, and we have to create innovative ways of early means to get through early clinical development. And remember back to that slide, providing confidence in the value early. So the thought is, the current state is reducing hospitalization rates, which happen maybe 1 to 3 times a year, or looking at the duration of hospitalization, acute chest injury or other organ failures, with no really validated biomarkers to teach you anything along the way. You're just waiting, you're doing either prophylactic and then you're hitting the hospital. What we're really trying to do in the future state again, it's this patient-centered endpoint. That we have this composite endpoint of the VOC day, which accounts for their their disease state and their decisions, regardless of what degree of medical utilization is being used, as well as then if we do collections of biomarkers that potentially, and also changes in activity that correlate with VOC endpoints. The goal is, as we accumulate enough data, could we actually be somewhat predictive of future VOC endpoints? There's the activity data, potentially with large enough data, could you actually create a profile that says you're at a higher probability of a future VOC state? Those are things we're always striving for. So the point is, by getting to this future state, we can then really look at the chronic therapy, because we're now capturing a larger number of events, 7 to 8 a year versus 1 to 3. So again, that's really important about using fewer subjects and shorter treatment periods, because if your alpha needs to be 45% or so, you do the math around cohort size. So you can maybe what needed to be 180 may only need to be 90 or 60 or so. So we have, that's one thing that's really driving this, and the biomarkers will better understand the nature of the VOC, as escalation over time, and potentially for new targets for intervention. And again, the predictive nature of either the EPRO or the biomarker changes. And again, registration timeline. So that could we work with the registry to say, this is now a co-primary. We're going to get 7 or 8 of these a year. We feel these are clinically meaningful. They're going to provide benefit. And so we can get the early pivotal studies faster, so that it's not such a daunting task to try to innovate and create new medicines for this patient population. In addition, by having a lot of those secondary things around fatigue, daily function, is there room here for differentiation between therapies? It isn't simply about the crisis itself, but it may be there are others about more of the pain or end organ damage. Could either a biomarker or actigraphy or a EPRO profile help provide that level of differentiation? So our group is actively working with, our outcomes group is working with developing a briefing document to go to the regulatory agencies to bring this forward as potential for co-primary. So as we progress, hopefully you'll be seeing this in future studies. And for any group out there looking to look at endpoints, this is not a proprietary tool. We'd love to have other groups be using this type of a VOC day endpoint. And it's about building that content validity. More data is better. So it's always a challenge to get that in place. But hopefully this has been informative, and a little bit about drug development, a little bit about going from point A to point B and getting ready for early clinical development. But thank you for attending, and we welcome any questions. Before I finish, though, I always forget to do this, and this is important. This is a lot. There's a lot of people, both within Pfizer, the rare disease, all of our collaborators on the studies, Boston Children's, the folks at Sanguine were very helpful as well. So again, this was a lot of good work by a lot of people, and so thank you everyone for attending.